Cell theory simply states that all living things are composed of cells, and that the cell is the smallest unit of life. Another commonly accepted part of the cell theory involves perhaps the most fascinating part of the study of cells. This part states that all cells come from other cells, and that process of cell division is mitosis. In unicellular organisms, like this amoeba, division produces two complete independent amoebas, just like the first one. In multicellular organisms, cell division permits an increase in the number of cells and permits the growth of the entire organism. In other words, each cell is not an independent organism. It's part of the whole. New cells in multicellular organisms are used for growth and repair of tissues. But what exactly is mitosis, or cell division, and how can one cell split into two identical copies of itself?
Although the entire division is one continuous process, biologists have divided mitosis into 4 visible phases during which certain events occur. Between divisions, the cell is in the stage called an interphase. Before any visible change takes place, the cell makes a copy of all of its genetic material. Then, in the first of the 4 visible phases, chromatin condenses into coils of chromosomes. Soon we can identify them as duplicate sets of chromosomes. The chromosomes then arrange themselves along what's called the cell's equator. The duplicate chromosomes then separate. An identical set moves to each end of the cell. The cell wall gradually forms in the middle, and this creates 2 new daughter cells with the same hereditary characteristics as the original.
Now let's watch mitosis according to its phases. During the interphase period prior to mitosis, the cell has been busy with the functions of its normal life, and in particular, with its own growth. During this period, the chromosomes aren't the rod-like bodies we normally associate with the word. Instead, they're in the form of a twisted mass of thread-like material called chromatin. At the end of interphase, the chromatin content doubles in preparation for division, but we can't see the doubling. Prophase is the first visible act of division, and it starts with the coiling of the chromatin to form chromosomes. As the coiling continues, the chromatin threads form into the conspicuous double chromosomes. Each set of identical twins is joined at a single point called a centromere. Now the nuclear membrane dissolves away. While all this is going on, an amazing structure of fibers begins to appear. They're known as spindle threads, and they run from one side of the cell to the other. A spindle thread is anchored to each centromere. You can imagine the spindle network as a kind of scaffolding on which the division will take place. Before we go on to the next phase, let's look at the prophase as it really takes place in a living plant cell. You can see the chromatin forming chromosomes. Then the nuclear membrane dissolves, and the cytoplasm and nuclear material freely intermingle. The fine threads are the spindle fibers. They're not easy to see in a living cell. Now the nucleoli disappear. At the same time, the twin chromosomes unwind. But they're still held together by the centromere. Up to now, the chromosomes have been scattered at random in the central part of the cell. In the next stage, the metaphase, they line up on the equator, ready for the division. The lineup is at a right angle to the spindle, and at the midway point between the two sides. The next stage, the anaphase, is actually the climax of the mitosis. Now, each centromere splits in half, and the two daughter chromosomes separate. Completely independent of one another, they will move apart. They're apparently pulled by the spindle fibers attached to the centromeres. The two identical sets of chromosomes move to the opposite sides of the cell.
Here is a blood lily cell in metaphase. The spindle fibers are slowly aligning the chromosomes along the equator of the cell. Now the anaphase. The daughter chromosomes separate, the spindle fibers pulling them to opposite sides of the cell. Here it is again. The chromosomes line up down the center. And the daughter chromosomes separate.
The final act of division is the telophase. In plant cells like this one, a cell wall begins to form, cutting through the spindle structure. The spindle fibers, guiding structures during the division process, now begin to disappear. While the cell is being divided in two, the chromosomes resume their original thread-like appearance, and new nuclear membranes form around them. Here's the actual telophase taking place. The cell wall begins to form, and the spindle disappears.
Now these are animal heart cells. You can see the duplicate sets of chromosomes emerging. The three dark nucleoli disappear. And the nuclear membrane dissolves. The chromosomes now line up down the center of the cell. Next, the anaphase, and the daughter chromosomes separate, each set moving to the opposite sides of the cell. In an animal cell, remember, there's no cell wall to cut the cell in two. Instead, the two daughter cells pinch together and pull apart. As the two separate, eruptions can be seen around the cell membrane. These are the result of physical and chemical adjustments that have to be made in the two new cells. Finally, the chromosomes mass together in each new nucleus, and mitosis is complete. This series of scanning electron micrographs shows an animal cell in division. The time required for such a division varies, depending upon the kind of cell. Some single-celled organisms can divide in less than 1 hour. Human skin cells divide in approximately 10 hours, and human brain cells don't divide at all.
Over 1 million different kinds of organisms have been described, each unique unto itself. Yet all of these organisms have basic characteristics in common. All organisms are composed of cells. These cells have many parts in common, and each common part has a similar function. These cells are produced from existing cells by mitosis, or cell division. Because of this cell division, organisms can grow and repair themselves, and life continues.